So the broader approach is trying to understand the biology and on the belief that, on the principle that this would lead to better patient management. And it's really very, I mean, to us, very pleasing, those of us interested in the biology of the disease, that agents targeting fundamental pathophysiological, biological processes rather than generic cancer-associated mechanisms have entered the clinical arena and have transformed the way we treat our patients. Ibrutinib is one such agent. Ibrutinib has revolutionized the management of patients, but there are still many open questions regarding the molecular mechanisms and the biochemical processes. So one of the projects we are presenting at the EHA meeting concerns a mechanistic study on the effects of ibrutinib at the translational and transcriptional level. I don't want to go into too much detail, but what we did in that work is utilize a novel, powerful, high-throughput methodology that is, let's say, between the RNA and the proteins. When you study the RNA, you have an idea of what was the RNAs that were inside the cell and the RNAs that are currently produced. When you study the proteins, you're studying the exact same thing. It would be interesting, though, to have a way to study which RNA molecules are translated into proteins under a particular condition. In our case, that condition is ibrutinib. So we want to see the changes that are induced by ibrutinib inside the cell, which proteins are actually translated, produced right now as we speak. So we utilize this powerful new methodology. It is called ribosomal profiling. So we analyze those RNA molecules that are translated into proteins right at the moment of the analysis. And we got some very interesting results pointing to new ways for improving the efficacy of ibrutinib. Now, ibrutinib has proven extremely effective across all biological subgroups of CLL, including, and that was a big positive surprise, cases harboring TP53 mutations. So now it is really wrong to say that these are doomed, because now we do have solutions. Ibrutinib and venetoclax are solutions for these patients. But should we consider all patients harboring a TP53 defect as equal? And there is evidence indicating that no, even within the TP53 aberrant subgroup, some patients are doing much better than others. And this is what we try to understand in the second project we are presenting, where we compared patients harboring a TP53 defect but experiencing a particularly mild clinical course, and versus those that are, let's say, typical for a TP53 aberrant profile. In other words, they progressed fast and were refractory to chemoimmunotherapy. And the nice thing that came out of this study was that the indolent ones, if you will, had a profile that is typical of cells, of B cells, that are not responsive so much to external drive, to triggers from the microenvironment. So we think that this work adds to the concept that the microenvironmental triggering is essential, in fact, the major drive for the evolution of CLL.